Alright guys, let's get started with Chapter 8. Hopefully the YouTube video was helpful in helping you understand the experiments that led up to the discovery of DNA as the genetic material. Our PowerPoint that goes along with our section, not as detailed, only 3 slides, and I will try to keep it short since you've already gone through it. But you know me, I get a little wordy sometimes, so I'll do my best. Starting with one of the most important things is that key concept, which I say at the start of every section. And the key concept here that you get is that DNA was identified as the genetic material through a series of experiments, not just one, not just two, but a series. And this essentially is what this section is about. It's going to run through each of those experiments with you. So let's take a look. Okay, and the first experiment discussed is Griffith, and Griffith was the one who discovered what he called a transforming principle. Now, Griffith wasn't trying to discover DNA, he actually did it by accident. He was actually experimenting with pneumonia, with the bacteria that causes pneumonia actually, and came across this transformation principle accidentally. As they explained, he used 2 forms of bacteria, and he injected these bacteria into mice. The S bacteria, or what were called the smooth bacteria, were deadly, and your R bacteria, which were the rough form of the bacteria, were not deadly. So what he found, in essence, was that when he injected the S bacteria into a mouse, the mouse died because it was deadly. It was virulent, was the word they used. If he injected the R bacteria, the mouse survived, and that's because this was the non-deadly form. He then took that S bacteria, which was the deadly form of bacteria, and he heated it first. And when he heated it first and then injected it into the mouse lived. So something had happened that had caused that bacteria, when he heated it, to not cause the mouse to die. He then took the heat-killed S bacteria and mixed it with live R bacteria. The R bacteria were the bacteria that were non-deadly. And when he mixed the 2 together, again, the mouse died. And this led him to believe that there had to be something that transformed that heat-killed S bacteria into a deadly bacteria once again when he mixed it with that live R bacteria. And he called this a transforming principle because he believed something transformed that caused it to change and kill the mouse once again. He didn't know what that was, but later they found out. So let's see what happens next. Alright, so the next scientist to come in was Avery, and what Avery did was he took Griffith's experiments and he isolated them. In other words, he took that transforming material that Griffith wasn't sure what was, and he performed some tests to figure out whether or not that transforming material was either protein or DNA. And he performed 3 different tests. He performed some qualitative tests, he performed some chemical tests, and he performed some enzyme tests. This little chart, shows the results of his chemical test. And here's how we would read this. We know that the value for DNA of nitrogen and phosphorus is this. We know that DNA is 15.32% nitrogen. We know that DNA is 9.05% phosphorus. And when we break that down into a ratio, we get 1.69. He took 4 different samples of this transforming material, and he determined that when he chemically analyzed those 4 samples, they all very much resembled the known values for DNA. So for our first sample it was 14.2, sample B 15.9, so on and so forth. So all of the samples very much resembled the known values for DNA, which again led him to believe that this was actually DNA and not protein. Finally, the enzymes test that he did, it says here that he did an enzyme test that showed only DNA degrading enzymes stopped transformation. Basically what that means is he would take an enzyme that killed DNA, and when he used that enzyme and killed the DNA, there was no transformation. So the transformation didn't occur. If he killed the protein if he used an enzyme that killed the protein, transformation still did occur, which again led him to believe that it was actually DNA that was the genetic material and not some kind of protein. And the last experiment was done by Hershey and Chase, and they wanted just to confirm that it was DNA. Once again, because, as scientists, we like to do things over and over and over again to make confirmation. It says here that Hershey and Chase studied viruses that infect bacteria. These are known as bacteriophages. Don't forget that bacteria are living things, so because they're living, they can also be infected with a virus. So what you see here in this picture is a little virus that is attacking a bacterial cell. It's kind of cool. They look like little spaceships that come in and land on the cells, and they literally inject their viral DNA or RNA into the cell. And what Hershey and Chase did was they tagged the viral DNA with a radioactive phosphorus. And what that means is it would make the DNA glow, essentially, if they were to take an X-ray. They also tagged the viral proteins, so the proteins of the virus, with radioactive sulfur. And in that case, it made the proteins glow when an X-ray was taken. So they allowed these viruses to infect the bacteria. And then they took the bacteria, they essentially put it into what is the same as a blender that you have in your kitchen. They smushed it all up, threw it into a centrifuge, and they analyzed it to find out what landed where. And essentially, when you centrifuge something, a centrifuge spins things down, like in our DNA simulation we did in class the other day with the extraction, and you put the tube into the centrifuge. When you do that, the heavier particles will fall to the bottom. So your heavier particles would fall down here, and then your lighter particles will stay up top. Proteins are heavy, so proteins will tend to fall, and DNA is much lighter. So the DNA would tend to stay on top of the vial after they spun it down. And what they found was the tagged DNA was found inside the bacteria every single time. So the fact that the DNA was inside the bacteria and that the protein was not inside the bacteria was conclusive that it was actually DNA that was being injected into the bacteria. So that's where that study came from. But anyway, hopefully you got some more information from that YouTube video, a little bit more helpful than the slides here, a little bit short, but again, not the most exciting section for this chapter. All right, guys, have a great weekend. Sorry I missed you today, and I will see you guys on Monday, and we'll make some DNA, what are they called? DNA models, paper models. That's what we're doing. Have a good weekend, guys. Bye.